One of the greatest tools we have is the power of observation. We have learned a lot by looking at the night sky, or watching how animals interact with each other, or looking at cells grow and divide. But how closely can we observe a cell? Could we take a peek inside and see what's happening at the exact moment a virus initiates an infection? With a specialized kind of microscopy called cryo-ET, we can. In the VIA lab, we are working to capture images of the HIV-1 virus just prior to membrane fusion to answer some important questions about how it is able to infect human cells. To do this, we use a specialized disc called a grid, add our cells, add virus, and then plunge freeze it in liquid ethane so that the viral fusion is frozen in time. We can put this frozen grid on a transmission electron microscope and collect a series of images from different angles. These images can then be used to reconstruct a three-dimensional structure that can provide new insights. The fight against HIV is an ongoing battle, so by better understanding the virus's structure during the initial fusion steps, we can discover its weaknesses and improve upon current treatments and preventative care. With such a powerful observation tool, our lab is able to investigate not only HIV, but many other biological questions. We study how certain phage-infected bacteria can have sophisticated cellular biology similar to eukaryotes, such as cytoskeletal filaments and a nucleus. We aim to discover the ultrastructure within tunneling nanotubes, an understudied cell-to-cell mode of communication. We want to visualize and understand structure of the nuclear pore complex, which transports materials between the nucleus and cytoplasm. We study the molecular landscape of an important enzyme, LRRK2, which has indications in familial Parkinson's disease. We want to investigate the role of condensates in the organization of the early secretory pathway in neurons. We want to understand how the protein that is responsible for how we perceive touch, piezo, mechanically functions in its native environment. And finally, we are working to build the tools to help find your proteins of interest within the cell. Thanks for watching!